Very good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, today we are going to start with our 100 topics for NEET 2026. That is, we are beginning from cell the unit of life, and from this the initial foremost important topic after cell theory and discoveries is prokaryotic cell. So today we are going to explore complete prokaryotic cell. Fine. So prokaryotic cell, if you look into first let us go from outer layer to the inner layer. Fine. Prokaryotic cell includes, like what are included under prokaryotes? You tell me what are included under prokaryotes. Prokaryotes include like mycoplasma, blue-green algae and all monerans Monera are the sole members of the prokaryotes. Now coming to the prokaryotic cell, first prokaryotic cell envelope we will look into. How cell is covered with, prokaryotic cell is covered with. Prokaryotic cell is enveloped by a cell envelope. Just a second. Prokaryotic cell is enveloped by cell envelope. Cell envelope has 3 layers in prokaryotic cell. Okay, it has 3 layers. It includes, first layer is glycocalyx, second is cell wall, third is cell membrane. These 3 layers together perform one function, that is giving protection to the cell. 3 of them are protective in function. But then when we look into each layer of the cell envelope, the outermost layer, that is peripheral layer, that is glycocalyx. Glycocalyx, if it is thickly composed of glucose and protein units, okay, if it is thickly composed of polysaccharides, then it is called as capsule. Okay, then it is called as capsule. And if the polysaccharide units are loosely arranged, then glycocalyx is called as slime layer. Okay, then it is called as slime layer. First we will look into what is the benefit that bacteria will get if the outer layer is capsule. If the outer layer is capsule, the bacteria can, if the outer layer is capsule, the bacteria can protect itself from host immune system. The bacteria protects itself from host immune system, protects from host immune system. If it is loosely arranged, we are calling it as slime layer, right? So if it is a slime layer, this bacteria will prevent itself from dehydration. Okay, if the outer layer, if the glycocalyx is loosely arranged, we are calling it as slime layer. If slime layer is the outermost layer, then this will prevent the bacteria, okay, from dehydration, or you can say prevents from loss of water and minerals, loss of water and minerals. Clear beta? So this is about the outermost wall layer of the cell envelope. Second is what? Cell wall. Cell wall in bacteria is composed of what layer? Peptidoglycan. Cell wall is composed of peptidoglycan. Based on the composition of the peptidoglycan, whether it is thickly present or lightly present, again we have classified into 2 types, that is gram positive bacteria and gram negative bacteria. Then about cell membrane. Cell membrane will be having some invaginations towards the cytoplasm. We call them as mesosomes. Okay, so this is about the cell envelope. So, cell envelope, the outermost layer is called as glycocalyx. If it is tightly arranged, it is capsule. It will protect the bacteria from host immune system. If it is loosely arranged, it is slime layer. It will prevent from loss of water and mineral nutrients. Second is cell wall. It is composed, bacterial cell wall is composed of peptidoglycan, that is protein and glucose units. So, based on the thickness of peptidoglycan, if it is very thick, about 90% of it is peptidoglycan, then it is gram positive bacteria. Just 10 to 20% of peptidoglycan is there in the cell wall, then it is gram negative bacteria. We will discuss in detail about that by doing gram staining, by discussing about gram staining procedure as well. Second and last innermost layer of the cell envelope is cell membrane. Clear beta? Next. So, now as I told you, based on the thickness of peptidoglycan, bacteria are classified into gram positive and gram negative bacteria. Okay? Gram positive and gram negative bacteria. Gram positive and gram negative bacteria. So, yes, gram positive and gram negative bacteria, if you see what this gram staining procedure is given by Christian Gram, okay, the scientist Christian Gram, okay, wherein he has taken two strains of gram positive and gram negative strain. Both the strains he has first added what? Crystal violet dye, okay, what dye beta? Crystal violet dye. The basic dye he has added is crystal violet dye. Okay, those bacteria which has very thick peptidoglycan has absorbed the dye totally, and those which doesn't have thick peptidoglycan has loosely absorbed that. After that, he has cleaned both the strains with alcohol and then with mordant, that is iodine. After that, discoloration is observed, decolorization is observed in gram negative bacteria because it didn't retain the color, okay, it didn't absorb the color totally. After that, he has restained with one more dye, that is safranin, okay, absorbing the safranin dye again, gram negative bacteria turned to pink color, whereas gram positive bacteria retained the first stain only, showing the crystal violet color, okay, that purple color, fine. So, this is about gram staining experiment and gram positive and gram negative bacteria. Let us now look into the cell wall of gram positive and gram negative, and let us also look into the differences between gram positive and gram negative bacteria. If you see gram positive bacteria beta, the cell wall is 80%, it is composed of what? Peptidoglycan only. The cell wall is 80% composed of peptidoglycan, just 10 to 20%, 10 to 15% to be frank, 10 to 15% it is composed of what? Lipid layer, okay. So, peptidoglycan is absorbing the stain completely, whereas gram negative bacteria, if you see, peptidoglycan is just 10 to 20%, whereas major composition of it is with respect to lipid bilayer, okay, it is with what ma? Lipid bilayer, okay. So, this is not retaining the initial stain, that is crystal violet, okay, and the major differences is gram positive is made up of murin. Gram positive bacteria has murin and teichoic acids, murin and teichoic acids, whereas gram negative bacteria doesn't have murin totally. So, we call it as pseudomurin, and teichoic acids are absent in the gram negative bacteria, in the cell wall of gram negative bacteria. Based on the composition, they are classified into gram positive and gram negative. Here one point to highlight is most of the pathogenic bacteria, are gram negative. They are what? Gram negative. Most of the pathogenic bacteria are gram negative bacteria. Clear ma? Fine, right? Next. So, here now we have come to the last layer, that is cell membrane. We have discussed about the glycocalyx, cell wall and its composition. Based on the composition of peptidoglycan, it is gram positive and gram negative. Now the lowest layer of the cell envelope or the close layer to the cytoplasm, that is cell membrane. As we have discussed, cell membrane has the invaginations. So these invaginations can be in the form of, vesicles or lamellae or tubules, vesicles, lamellae or tubules. So it can be in the form of vesicles, it can be in the form of lamellae, it can be in the form of tubules. Towards what? Towards cytoplasm. It is towards, these invaginations are towards cytoplasm. Clear? So this invaginations of cell membrane, in the, whether it is in the form of vesicles, lamellae or tubules towards the cytoplasm, what do you call it as beta? Mesosomes. Fine. Same invaginations in BGA, okay, blue green algae, or you can also say it as cyanobacteria. Same invaginations in blue green algae or cyanobacteria, those invaginations are called as chromatophores. Those are called as what? Chromatophores. Chromatophores. Fine. They absorb the sunlight and they conduct the photosynthesis as well. So these are colored pigments, chromatophores in cyanobacteria, whereas in normal bacteria it is mesosome structures. Clear, right? So here you can see the cell wall and cell membrane, mesosomal invaginations into the cytoplasm and mesosomal sacs also. So how these invaginations are in the form of tubules. Now let us look into the functions of the mesosomes. What are the functions of the mesosomes? Functions of the mesosomes include, okay, functions of the mesosomes include, most important beta take down. First is cell wall formation, second DNA replication in prokaryotic cell. Most important function that is respiration, secretion and increase in surface area, So here you have to see, it leads to the cell wall formation of the bacteria, DNA duplication of the bacteria, most importantly respiration. How respiration? By releasing out the respiratory enzymes, into the cytoplasm. As they are conducting the respiration in prokaryotic cell, there is a most important statement that mitochondria of prokaryotic cell, okay, mitochondria of eukaryotic cell, is equivalent to mesosomes of prokaryotic cell. Clear? This is the most important statement. Remember, many a times this is asked in statement based questions beta. So mitochondria of eukaryotic cell is equivalent to mesosomes of prokaryotic cell because it is respiratory in function, and it secretes secretion, and because of those invaginations towards the cytoplasm of those cell membrane, this increases the surface area of the cell. Clear? These are the functions of mesosomes. Now entering into the cytoplasm, first we will look into the genetic material, okay, genetic material inside the cytoplasm. Fine. Genetic material inside the cytoplasm. When it comes to genetic material, prokaryotes will have two types of genetic material. Here you can clearly observe One is genomic DNA and the other is plasmid DNA. Genomic DNA is also called as, okay, genomic DNA is also called as cytoplasmic DNA. Genomic DNA is also called as chromosomal DNA, sorry, genomic DNA is also called as chromosomal DNA, fine, genomic DNA or chromosomal DNA, one and the same, whereas plasmid DNA is also called as, plasmid DNA is also called as non-genomic DNA or extra chromosomal DNA. This is called as non-genomic DNA or extra chromosomal DNA. Fine. Genomic DNA, as we all know, this maintains or conducts the, looks after complete metabolic activity of the bacteria. Okay, this looks after complete metabolic activity of the bacteria. Whereas plasmid DNA, okay, plasmid DNA is helpful in what? What are the functions of plasmid DNA? This gives particular phenotypic character to the bacteria, resistance to antibiotics, okay. This gives what? Phenotypic character to the bacteria, specific phenotypic character, okay, specific phenotypic character, to the bacteria, and antibiotic resistance, and most importantly it helps in sexual recombination, okay, sexual recombination, that too conjugation process. What process beta? Conjugation. Clear? Each and every point is important, I am telling you, okay. When it comes to genetic material, bacteria is having two types of genetic material, genomic DNA, which is also called as chromosomal DNA, and plasmid DNA, which is also known as non-genomic DNA or extra chromosomal DNA. And this genomic DNA or chromosomal DNA will take care of complete metabolic activity of the prokaryotic cell, whereas plasmid DNA has unique functions, that giving specific phenotypic character, antibiotic resistance, and acting and promoting the conjugation in sexual recombination. So, this is about genomic and non-genomic DNA. Now, other non-membrane bound organelles in the prokaryotic cell. We all know that prokaryotic cell does not have membrane bound nucleus and they do not have membrane bound organelles, but prokaryotic cell do have non-membrane bound organelles, okay, non-membrane bound organelles. Non-membrane bound organelles include, okay, non-membrane bound organelles include ribosomes and storage organelles, okay. Reserve food material is stored in inclusion bodies. This does not have any membrane, okay. So, now in ribosomes, what type of ribosomes are present? Ribosomes are 70S type of ribosomes, wherein larger subunit is 50S and smaller subunit is 30S. Smaller subunit is 30S, larger subunit is 50S. This 70S type of ribosomes are present. Where are these ribosomes present? Okay, if let us suppose this as a cell, okay, this as a cell and this is the cell membrane, okay. Ribosomes are present attached to the cell membrane and inside the cytoplasm, okay. Ribosomes are present attaching to the cell membrane and inside the cytoplasm. Those ribosomes which are present attaching to the cell membrane are helpful in protein translocation from cell to cell, and those ribosomes which are present inside the cytoplasm are helpful in protein synthesis, okay. They are not involved in translocation of proteins. Clear with the functions of these two ribosomes? One more specific feature about ribosomes that I have to highlight here is, At the time of protein synthesis, over the mRNA, ribosomes are present. In eukaryotic mRNA, single ribosome is present, which is called as monosomes. But in a prokaryotic cell, many ribosomes are present over the mRNA. Many ribosomes are present. This is called as polysome. Polysome, also noted as polyribosome. Okay, also called as polyribosome. Fine. So this single mRNA having multiple ribosomes over it will give you polycistronic expression, that is multiple protein expression is observed, okay, which is polycistronic expression. Fine, ma. Fine. So here what type of ribosomes are present? 70S ribosomes are present, larger subunit is 70S, smaller subunit is 30S, and single mRNA having many ribosomes on it, this is polysome or polyribosome condition, and this gives multiple protein expression, which is called as polycistronic in expression. Fine. This is the complete information about ribosomes in a prokaryotic cell. Coming to reserve food material, right? They will be storing reserve food material. So inclusion bodies are of many types. Inclusion bodies are of many types. Phosphate granules, cyanophycean granules, and sulfate granules. These are different types of stored materials or reserved food materials that are present in the form of inclusion bodies in a prokaryotic cell. This is about non-membrane bound organelles in the prokaryotic cell. Fine. Now if you see beta, next, so I have highlighted about non-membrane bound organelles, and here you can clearly look into the polysome or polyribosome condition. Okay. Next, based on the shapes of the prokaryotic cell, they are classified into different shapes, okay, like cocci, which is having circular shape, fine, bacilli rod shaped, spirillum spiral shaped bacteria, vibrio comma shaped. Based on the shapes, they are classified into 4 types. What are they? Cocci, bacilli, spirillum and vibrio. Okay, spherical, rod shaped, spiral shaped and comma shaped bacteria. And now this is the diagram which is given in our NCERT on sizes of the cell, which is very important. Okay, eukaryotic cell has a diameter around 10 to 20 micrometers in diameter. Okay, eukaryotic cell has 10 to 20 micrometers in diameter. Bacteria, typical bacteria will be having 1 to 2 micrometers of diameter. PPLO, that is pleuropneumonia like organisms, comes under mycoplasma, which are the smallest living cells, is about just 0.1 micrometers. Next is virus. Virus is from 0.02 to 0.2 micrometers in diameter. Fine. This is about different sizes of the cell. Fine, ma. So once let's quickly look into whatever we have learnt today. Yes, we have looked into the complete prokaryotic cell, and now I would like to complete this segment by highlighting you about the motile and non-motile structures also. Okay, motile and non-motile structures of prokaryotic cell. Motile and non-motile structures. So yes, if this is a bacteria, let us consider, okay, let us consider this is a bacteria which is having cell wall and cell membrane, and inside it has cell membrane. Okay, so this is cell membrane, this is cell wall, and this is outer layer, let us suppose capsule. Fine. From this, motile or non-motile structures, they originate from cell membrane and they are extensions of the cell wall. Let us consider this as flagella. Okay, let us consider this as flagella only. Fine. And inside what type of genetic material do we have? Genomic DNA, which is in a particular location, that is called as nucleoid. And what type of DNA is it? Double stranded circular DNA. And here plasmid DNA. Next is what? Plasmid DNA. Plasmid DNA is also double stranded circular DNA. Fine. This is helpful in conjugation and phenotypic character and resistance to antibiotics. And inside you have ribosomes in the cytoplasm and attaching to the cell membrane as well. Fine. And then you have inclusion bodies as well. Most importantly, the extensions of cell membrane, those are what? Mesosomes. Fine. Now coming to the motile structure, that is flagella. Flagella is the only motile structure in the bacteria. Do remember, flagella is the only motile structure in prokaryotes. And flagella is having what type of protein in the prokaryotes? Flagellin. Okay, this has what protein ma? Flagellin. And what is the arrangement, microtubule arrangement in the flagella? What is the microtubular arrangement? 9 plus 0. 9 plus 0 arrangement is seen in the prokaryotic cell. Remember that. What are the parts of the flagella here? It has basal body, next is hook and filament. Basal body, hook and filament are the parts of flagella, and flagella is having protein flagellin. It has 9 plus 0 arrangement. Originates from cell membrane and these are the extensions of cell wall. Coming to non-motile structures, two non-motile structures are present over the bacteria. Those non-motile structures include, just a second, cilia and flagella. Non-motile structures include, just a second, pili and fimbriae. Okay, it includes pili and fimbriae. These are the non-motile structures which are present over the surface of the bacteria. These are also cell wall extensions. So this which I have drawn, these are what? Fimbriae, and this is structures which I have noted here. These are what? Tubular extensions, pilus. How you can describe fimbriae? Okay, fimbriae are bristle-like structures that are sprouting out of the cell wall. What are fimbriae beta? Bristle-like structures that are sprouting out of the cell wall are called as fimbriae. Fimbriae are only helpful in attachment to the substratum or to the attachment of the host cell. Fine. Whereas pilus are tubular extensions. Pilus are what? Tubular extensions. Tubular extensions that are arising from the cell wall of the bacteria. Pili has two functions. One is attachment to the host cell or the substratum or promoting cell to cell attachment. Second is pili helps in sexual recombination, that is pili helps in sexual recombination, that is conjugation process. Okay, pili helps in conjugation, where you call it as sex pilus. Okay, sex pili or sex pilus. Fine. So this is about the non-motile structures and motile structures. With this we have discussed completely the structure and functions of the prokaryotic cell. Let me complete this session by giving you a hint on how to learn the metabolic diversity, that is nutrition, respiration and reproduction, that is both asexual reproduction and sexual recombination we say. Just now we have learnt, we have heard about the term conjugation. More two types of sexual recombinations are present. Those are transformation and transduction. This complete information you will learn under Monera of the chapter biological classification. So, whenever you are learning prokaryotic cell beta, prokaryotic cell from the cell the unit of life, it is connected with the Monera kingdom of biological classification. There you will get complete details about the metabolic diversity and the reproduction as well. So, with this we are done with the prokaryotic cell. Any doubts anywhere from the beginning of the class to the end, you can ask me and you can drop in the comment section. Any doubts anytime. Okay, I will be answering all your doubts, ma'am. So, this 100-day session is really helpful where we are going to cover the complete concepts. Okay, complete concepts are covered and highlighted of the NEET syllabus. This is really helpful. So, yes, stay tuned and look into all the classes of this 100 days challenge. Fine? Yes, thank you.